Here we describe a protocol employing immunofluorescence, confocal microscopy, and computer-based analysis to determine the distribution of a synaptic protein relative to a reference protein. Our method circumvents the inherent variability of immunofluorescence stainings by yielding a ratio rather than absolute fluorescence levels.
Additionally, with this method, you can analyze the heterogeneity of protein distribution on different levels, from whole brain slices to brain regions to even subregions, such as the different layers of the hippocampus. This technique can be adapted to determine the protein distribution in different tissues other than the brain and model systems other than mice.
Begin by washing previously isolated and fixed mouse brain as described in the manuscript. Then transfer the brain to a 50 milliliter reaction tube with 30% sucrose in 0.1 molar Pb. To cryoprotect the brain, incubate it at 4 degrees Celsius for 48 hours or until it sinks. After that, use a sharp blade to trim the cryoprotected brain depending on the area of interest. Then place the brain in a cryomold and add optimal cutting temperature compound to embed it, making sure to avoid bubbles, and orient the brain properly so as to have a coronal plane of cutting. Place the cryomold in the -80 degrees Celsius freezer until it is frozen solid. Mount the frozen tissue on the cryomicrotome and wait for at least 15 minutes before sectioning to equilibrate it to the microtome temperature. Start cutting the brain into 25 micrometer thick coronal slices.